This is the second video for Labs 3 and Labs 4. So now on to a plant cell, the onion epidermis. So let us kind of draw what that looks like. So unlike the circular-ish animal cell, you can expect to find plant cells to be very linear, very geometric, much more rectangular. So let's take a note of that. So these cells are rectangular. You can think of them as more organized. Okay, so what I actually just drew, that isn't the cell membrane, that is the cell wall. This right now is the cell membrane, what I just drew. Okay. And these are also granular, the onion cells. And we can also expect to find a nucleus. Okay, so let's label this for an onion cell. This is actually a cell wall. And these are found in only plant cells. Now this right here, this is actually the cell membrane. In its normal environment, it's hard to distinguish the border of the cell membrane, but there is a way to isolate it, which we'll talk about later. Again, any region inside the cell membrane, this is the cytoplasm. And in this case, it happens to be granular, so we can give it this additional name, a granular cytoplasm. And then lastly, we have a nucleus. So this onion epidermis, it is a plant cell, but this one does not have any chloroplasts. So you will not find any in this structure. That's because these are usually underground where there is no sunlight, so it doesn't make any sense for them to have any chloroplasts. Okay, now how do we kind of isolate the cell membrane from the cell wall? So we can do that by placing this in a hypertonic solution. So let's see what we can expect to see. So on the left is going to be the cell in its normal environment. So we'll say in its natural environment. But on the right is going to be one placed in a hypertonic solution. Hypertonic solution. What that basically means is that the water molecules in the cell membrane, once placed in the hypertonic solution, are going to leave the cell membrane. So these waters are leaving. And you can expect the cell membrane to shrink in size because the water left. So H2O blue circles, H2O left the cell membrane. Okay, so now we can clearly see the difference between cell wall and cell membrane. Oh, yep, that's a cell wall. Oh, you can clearly see the cell membrane now. But everything else is still the same. Okay, so here is an image of the cheek cell next to a onion cell, and hopefully you can see the difference between these two structures. Okay, next plant we're going to look at is something called spirogyra. And this is basically long rectangles put together like so. And unlike the onion plant cell, this one does have chloroplasts. But this one has an interesting shape. It's in a spiral shape, which is why we call this spirogyra. One reason. So this is actually the chloroplast. This organelle has this interesting shape. So this is the chloroplast. And the chloroplast is responsible for a process called photosynthesis. This is where photosynthesis occurs. So if you remember in photosynthesis, this is where we take carbon dioxide. So that looks like this. And it mixes with water. We know water looks like this. And it's going to form a carbohydrate that we know, and this guy here is glucose. And it also forms oxygen, O2. And O2 looks like this. So photosynthesis creates glucose and oxygen. I forgot to mention on the left side, we also need energy input. So one of the reactants here would be energy in the form of light energy, which we call photons. Call them photons. So these chloroplasts up here are making glucose. And if you take glucose and you put a bunch of them together, so let me take my product of photosynthesis. Here is a glucose. There is another glucose. Here is another glucose. If you were to add these together, they will form starch in the plant cell. And the starch is actually stored in these structures in the chloroplast called pyrenoid bodies. Use a different color. Let's use this color. So these structures, these guys are called the pyrenoid bodies, and they store the starch that was just made. So pyrenoid body. And they store starch. And now you look at this cell on its own, but then when you add iodine to it, remember iodine stains starch, so we should see some reaction or change in color. So remember that iodine or Lugol's, reacts with starch. These guys here are the products, these glucoses, products of photosynthesis. Photosynthesis. Okay, and lastly, these do have nuclei, but they might be hard to find. But they do have nuclei. It might be transparent, but it's there. So, nucleus. Okay, so there is some spirogyra. Okay, the last plant cell you are going to look at is elodea. So here's what this looks like. Very similar to onion cells, but these are much more greener. And again, the cell wall is hard to differentiate against the cell membrane. So first off, one main difference is that the chloroplasts here are not like the ones in spirogyra. These appear as little circles throughout the entire molecule. So these are the chloroplasts. These are the chloroplasts. But everything else is still the same. This is your cell wall, unique to plants. This border right here, this would be your cell membrane, the border of the cell membrane. Again, any region on the inside, we call this just cytoplasm. And then for this case, we can't really see the nucleus because it is colorless and transparent, so you don't have to worry about finding that in this structure. Okay, so now if I were to place if I were to place my elodea in a hypertonic solution, I'd be able to see the clear distinction between cell membrane and cell wall, just like for the onion cell. So again, left is going to be one in its natural environment, where there's no osmosis, no diffusion occurring. Let me draw in my chloroplasts. Let me draw in my chloroplasts. Okay, so we will say natural environment on the left side. And on the right is the same cell but placed in a hypertonic solution. Hypertonic solution. So again, in the cytoplasm, we have water molecules, which I'll represent as these blue circles. So blue circles are H2O. Blue circles are H2O. So when you place this in a hypertonic solution, water is going to exit the cytoplasm. So water is exiting. As a result, oops, I should have not drawn this in. As a result, your cell, the cytoplasm and cell membrane is going to shrink. But everything else that was on the inside is still there. So there are my chloroplasts still. So we're going to say H2O is going to leave the cytoplasm. Sorry, leave the, yes. So now we can say here two things. You can say that both the cell membrane, you can see it now actually, both the cell membrane and the cytoplasm shrunk. Both of them shrunk. And now you can clearly tell where the cell membrane is in relation to the cell wall. So here is elodea. Here's a picture of elodea. And that's it for this lab.